Okay, so let's look at the events that takes place at the neuromuscular junction. And these are very similar to what takes place at the neuroeffector junction, all right? But there are some important differences. So this is our motor neuron. Okay, this is our axon terminal. And then this is the motor end plate. The motor end plate is folded like this to allow the increase in surface area and the transmission of this signal. So this is the motor end plate. And this is on the skeletal muscle side. Now within these, within the axon terminal here on the neuromuscular junction, on the motor neuron side, are synaptic vesicles. We looked at this before. And within those synaptic vesicles are our neurotransmitter, and in this case, it's only acetylcholine. Okay, only acetylcholine is found at the neuromuscular junction. Okay, so I'll put acetylcholine here. On the presence of the motor end plate, we have two important structures. One is going to be the receptor. So we have our receptor here. And that's going to bind our neurotransmitter. And then we also have another enzyme called acetylcholinesterase. Acetylcholinesterase. And we're going to talk about that function. So this is our nicotinic receptor. And you want to make a note that this is ionotropic. And we have acetylcholinesterase, which is this important enzyme. We'll talk about acetyl. Okay, so let's talk about the steps of what takes place here. So first of all, an action potential, depending on the summation of IPSPs and EPSPs, an action potential is fired in this neuron. It travels down the axon terminal, and it arrives at the terminal bouton. Now, when it gets to the terminal bouton, it's going to trigger the opening of voltage-gated calcium channels. These voltage-gated calcium channels are going to open up, and we're going to have the influx of calcium here. Okay. Calcium comes into the terminal bouton, and that triggers exocytosis of these vesicles, and this dumps all of this neurotransmitter into the synaptic cleft here. Okay. The neurotransmitter is then going to bind onto this nicotinic receptor. And because this is an ionotropic receptor, we're now going to have the opening of this channel and the influx of ions. We're going to have the movement of ions here, sodium coming in and potassium coming out. All right, so sodium is coming in more so than potassium goes out. And so that is going to depolarize the motor end plate and result in an end plate potential, which will allow this muscle to contract. Now, in order to cease muscle contraction, we're going to have to clear this neurotransmitter from the neuromuscular junction. All right? Otherwise, you would have that muscle continually contracting, and that's not what we want. That's going to be spasticity or rigidity. So in order to prevent that, we have to clear away the excess neurotransmitter when this muscle contraction is over. In order to do so, acetylcholinesterase is the enzyme that breaks down this acetylcholine into two molecules. It's going to break it down into choline and acetate. Okay, acetate can diffuse away from the junction, and choline is going to be reuptaken into the axon terminal and recycled to make more acetylcholine. Right, so this is your choline coming back into the neuromuscular junction. I'm sorry, back into the terminal bouton to be recycled to make more acetylcholine. Okay. So that's the importance of acetylcholinesterase. We sort of vaguely talked about this uptake enzyme earlier, and this is its specific job and how it functions. We're going to see when we look at some clinical examples later on. Any questions on this? Any questions? So the contraction of the muscle or the excitation of the muscle fiber is called an end plate potential. And that's what's brought about when we excite this muscle fiber by opening up these nicotinic channels, and that happens when acetylcholine binds to this receptor. All right, any questions here, guys? Okay, so let's continue on. Okay, so these steps are what we just went through, and they pretty much summarize what takes place at the neuromuscular junction between the action potential arriving at the terminal bouton and then finally muscle contraction in the form of an end plate potential. Okay, two things we want to point out is that the terminal bouton has synaptic vesicles that only contain acetylcholine. That's the only neurotransmitter here. And the only receptor is your nicotinic receptor. There are no metabotropic receptors involved in the neuromuscular junction. So we do not see muscarinic receptors here. We do not see adrenergic receptors here. Only nicotinic cholinergic receptors. As well as the presence of this enzyme, acetylcholinesterase, which is basically taking excess acetylcholine that's left at the junction after muscle contraction, breaking it down into acetate and choline. Choline is reuptaken into the terminal bouton and recycled to make more acetylcholine. Acetate and any excess acetylcholine can be diffused away from the junction.